Okay, in this video we're going to talk about cytoplasm. And remember, cytoplasm is really all the cellular material that's located between the plasma membrane and the nucleus. And cytoplasm is made of three major things. We've got cytosol, which is the gel-like solution of water, insoluble molecules like proteins, salts, sugars. We also have cytoplasmic inclusions, which are basically insoluble molecules that can vary based on the cell type. But some examples of different types of inclusions you can find in cells are glycogen, different types of pigments like melanin, or lipid droplets, vacuoles, and even crystals of different substances. Now, the organelles are really what we'll focus on for the majority of this video. And remember, organelles mean tiny little organs. They're basically metabolic machinery and structures of the cell. Each organelle having its own specialized function. And there's different types of organelles. We have the membranous or non-membranous organelles. And we're going to talk about those organelles for the most of this video here, where membranous organelles include mitochondria, endoplasmic reticulum or ER, Golgi apparatus, peroxisomes, and lysosomes, each of these having their own functions. And then the non-membranous organelles including ribosomes, like free ribosomes, cytoskeleton, and the centrioles. Now, the membranes of our organelles allow for compartmentalization, which is crucial for cell functioning, because often what goes on inside of the membranous organelles you wouldn't want necessarily just to be leaking out inside of the cytoplasm. So it's important then to compartmentalize these functions in order to maintain stability and structure within the cell. Now, mitochondria is the first membranous organelle we'll talk about. Remember, this is like the power plant of your cells because they produce most of your body's ATP. Now, mitochondria produce most of this ATP through aerobic cellular respiration, and this is oxygen requiring, therefore. Now, the mitochondria are membrane bound because it has a double membrane, an inner membrane that has many folds called Cristae, and these are embedded with proteins that play a role in cellular respiration. If you all remember going back to previous biology classes, you probably learned about the electron transport chain, but this is located within the inner membrane. Now, mitochondria actually have their own DNA and RNA, as well as their own ribosomes. In fact, mitochondrial ribosomes are more similar to bacterial ribosomes than they are of other eukaryotic ribosomes within our cells. So it's believed then that mitochondria may have actually started out as their own kind of unique bacteria, because they even divide through fission, which is how bacteria divide. So it's likely that what would become a eukaryotic cell is basically a symbiotic relationship between an intracellular bacteria, which we now call mitochondria. Now, remember, the mitochondria is kind of this little bean-shaped organ here, or organelle rather, and you have two membranes, an inner membrane and an outer membrane. The inner membrane is studded with lots of proteins that are involved with cellular respiration, and this is like the power plant of your cells because it essentially helps to convert chemical energy from other molecules to a usable form of chemical energy, which is ATP. And these mitochondria are extremely active. In fact, you produce your own body weight in ATP every day. But the reason why that works is that you recycle that ATP, many times. Now, ribosomes are these non-membranous organelles that are the site of protein synthesis. Now, mitochondria have their own ribosomes, but there's two different types of ribosomes. We got free and membrane-bound ribosomes. The free ribosomes are freely floating protein, which are the sites of synthesis of proteins that you find in the cytosol or in the membranes of other organelles. But we also have membrane-bound ribosomes, which you find on rough endoplasmic reticulum, which is what gives it its kind of rough appearance. This is the site of synthesis of proteins that are going to be incorporated within like the membranes of your plasma membrane, or lysosomes are also created by rough endoplasmic reticulum, or proteins that are going to be exported from the cell through like exocytosis. So we're going to talk about endoplasmic reticulum here. Remember, there's two major types, rough and smooth, and it consists of really a series of parallel interconnected cisterns that are made of flattened membranous tubes. That's essentially like a plasma membrane of sorts. Now, this endoplasmic reticulum is actually continuous with the nuclear membrane. This is important because genetic information is transported from the nucleus over towards rough endoplasmic reticulum, which is one of the sites of protein synthesis. So if you want to look at this endoplasmic reticulum, you'd see that that's what's shown in blue here, and you can see that it surrounds the nucleus and it's continuous with the nuclear envelope, so that when genetic material leaves the nuclear plasm, into the cytoplasm, we find that it actually can float over and then reach, rough endoplasmic reticulum, and then basically help to synthesize new protein. Now, rough ER is basically rough because it has lots of ribosomes. It's the site of protein synthesis, but specifically proteins that are going to be secreted, secreted either into the plasma membrane or secreted out of the cell. And, many of the plasma membrane proteins that are incorporated there in the membrane itself are actually made by this rough endoplasmic reticulum. Now, proteins enter cisterns as they're synthesized, and what's interesting too is they're modified along the way. So while these proteins are synthesized, there are other enzymes within rough ER that can modify those proteins in ways that make them, functionally acceptable for the cell. Now, the final proteins actually enclose in a vesicle, and this vesicle is sent to the Golgi apparatus for further processing. Smooth ER differs from rough ER because it doesn't have ribosomes studded on its membrane. Rather, it's a network of looped tubules that are continuous with rough ER. And what you find are a variety of enzymes in the membrane of smooth ER, but not ribosomes. Now, these enzymes in smooth ER play a really wide variety of roles, things like lipid metabolism, so you can actually have cholesterol and steroid hormone synthesis here. You can also make lipids for lipoproteins, which is one of the ways that you transport lipids in your bloodstream. These are actually made by your smooth ER, as well as absorption, synthesis, and transport of fats within your cell. Smooth ERs also plays an important role in detoxification of chemicals like drugs and pesticides and different metabolites. So what's interesting is that smooth ER correlates with drug tolerance. So if someone can tolerate a drug very well, like a certain chemical, then they're going to have a lot of smooth ER to help break down that chemical rapidly. Interestingly enough, too, smooth ER plays a role with converting glycogen into free glucose. So it helps to break apart glycogen granules within the cell to free glucose, which then can travel around and can be used for chemical energy to make ATP. And smooth ER also helps to store and release calcium. It turns out that the fluid environment within the membranous sort of cistern of smooth ER is really rich in calcium. Now, calcium plays an important role in cell signaling, but it's especially important in skeletal and cardiac muscle cells because this smooth ER can actually help release calcium when you want skeletal and cardiac muscle cells to contract, because we'll learn about in future chapters how calcium is a stimulus for muscle contraction. Now, the Golgi apparatus is this stacked and flattened membranous cistern. Remember, it modifies and concentrates, packages proteins and lipids, and it receives these from rough ER. Remember, rough ER packages its contents into basically vesicles that then are transported to the Golgi apparatus, and there's three major steps here. The transport vesicles from ER actually fuse with the interface of the Golgi apparatus, and those proteins or lipids are taken inside and then therefore further modified, tagged, sorted, and packaged. You might wonder, how are they modified? Well, those proteins can be cut up in a variety of ways. You can actually attach other molecules to those proteins. They may be tagged for further processing or sorted in a variety of ways so that some proteins go to one side of the cell and other proteins go to another. And then possibly even repackaged for transport to other parts of the cell or for secretion. Golgi is also traffic director. So when you talk about tagging and sorting and packaging, the Golgi apparatus helps to direct the traffic of certain proteins within the cell, and this is done when those proteins are basically packaged into a new transport vesicle, which pinches away from the outer face or transface so that those contents are repackaged into a vesicle. So what this looks like then is you have this transport vesicle from rough ER. It fuses with the Golgi membrane here. You get processing of those materials within the cistern, and then those materials are repackaged within a new vesicle on the transface, and then those particular vesicles then can be transported either within the cell or can be sent to the plasma membrane for exocytosis and secretion. So an example of this could be something like insulin. So insulin is actually made by rough ER. It's packaged into a transport vesicle. It fuses with the Golgi membrane here. Then insulin is processed in a way that allows it to become ready to be activated. It's repackaged into a vesicle and then exocytosed out of the beta cells of your pancreas. That way insulin can actually leave the cell and enter your bloodstream. So this is just showing that Golgi secretory pathway where you see that, remember, packaged protein from the rough ER is sent to Golgi for processing, and there's a variety of things that can happen here. For one, it can actually lead to exocytosis of molecules, but two, it can also be sent to a phagosome, which can actually help to break down certain substances. There's also a third pathway here where vesicles can be incorporated into the plasma membrane itself. So we're saying that plasma membrane proteins can be processed within the Golgi. They're packaged within the membrane of this vesicle, so when the vesicle fuses with the plasma membrane, it becomes one with the membrane. So this is how those cell adhesion molecules or receptors all get in the plasma membrane from the start, is that they're basically secreted through a vesicle into the plasma membrane itself. Now, peroxisomes are basically these membranous sacs that contain powerful detoxifying substances. They're used to neutralize toxins like free radicals. And basically, if you didn't neutralize these free radicals, they would actually wreak cellular havoc all around the cell, which can damage your cell and lead to accelerated aging and decline. But the two major detoxifiers here are oxidases, which use oxygen to convert toxins and hydrogen peroxide, as well as catalases, which convert hydrogen peroxide back into water. That way this hydrogen peroxide doesn't damage your cells. Now, these peroxisomes also play a role in the breakdown and synthesis of fatty acids through these oxidation reactions. Now, lysosomes are similar to peroxisomes, but they have a different origin. It turns out these lysosomes actually come from the Golgi apparatus, and they're spherical membranous bags that contain digestive enzymes or hydrolases, and these are considered safe because they isolate the potentially harmful digestion from the rest of the cell. So just think about this would be sort of like the fire pit of your cell, because you can contain fire, which can break down things in a particular location. Now, these can be used to digest bacteria, viruses, toxins, and they help also degrade non-functional organelles. Some of the metabolic functions of lysosomes are to break down and release glycogen or break down and release calcium. And what's interesting too is that these lysosomes can be used for autolysis, where cells essentially kill themselves once they are irreversibly damaged or have undergone too much decline in age. And so lysosomes can actually digest the cell from the inside out. Now, you can see here these lysosomes, and you can see basically light green areas are where materials are being digested. And it makes sense how you'd want to compartmentalize digestion within these little vesicles here, because you wouldn't want these really strong or potent enzymes out in the cell, which could potentially degrade other organelles or even genetic material. Now, the cytoskeleton is actually an elaborate network of rods that run through your cytosol. It's hundreds of different kinds of proteins that link these rods to other structures. And you think of the cytoskeleton, it's basically like the bones of your cell, right? Because it plays a role in movement as well as support for the cell itself. And there's three major types of cytoskeletal elements. We got microfilaments, intermediate filaments, and microtubules. Now, the microfilaments are the thinnest of all these cytoskeletal elements, and they're made of semi-flexible strands called actin. Now, this causes a pretty dense web within your cells, and it actually helps to also strengthen the cell surface to resist compression of those cells. Some microfilaments are involved in motility or cell movement. So we're saying that when cells change their shape, like with endocytosis or exocytosis, microfilaments play a role. And you also find them in muscle cells because they play a role in muscle contraction. So here's these microfilaments. You can see that they're relatively small, and you can see they form a pretty dense web all throughout the inside of the cell, but they help support the plasma membrane, and some of these are actually involved in helping to change the shape of cells with things like transportation of materials or movement of cells as well. Now, intermediate filaments have an intermediate size, hence the name, and they're tough, insoluble, rope-like protein fibers that are made of 4 fibrils that are twisted together. They actually are involved with helping to resist pulling forces. So we find that these filaments attach to desmosomes. Remember, desmosomes were the intercellular junctions between cells. And these intermediate filaments, by attaching to desmosomes, help to hold those cells together very well. Some intermediate filaments have their own special names, like neurofilaments in your nerve cells, or keratin filaments in epithelial cells are all examples of intermediate filaments. So we see that, remember, it's a tetramer, which means you have 4 rope-like substances that are all interwoven. And you find that actually these intermediate filaments are also pretty widely distributed throughout cells, And like the microfilaments, they play a role in helping to stabilize the cell itself. But these intermediate filaments are actually attached to desmosomes, which could be involved with intercellular joining. That way it holds these cells together very strongly. Now the microtubules are the largest of your cytoskeletal elements. They consist of hollow tubes, and most of these radiate from the centrosome, which is basically like the microtubule organization center of your cells. Microtubules determine the overall shape of the cell and the distribution of organelles because your membrane-bound organelles are actually attached to microtubule networks. So many organelles are tethered to these microtubules to help keep them in place, and substances are moved along microtubules through motor proteins, which basically serve as like railroad tracks to help move material within the cell itself. So if you look at the microtubules, you find they're the largest of your cytoskeletal proteins, and they're all kind of organized here near the centrosome. And you can see that they radiate out away from that, so that other organelles are actually tethered to this whole network of track. And also vesicles can be transported along these tracks to different parts of the cell. Now, motor proteins are basically little proteins that can actually function in motility and movement of organelles and other substances around within the cell. Microtubules, are the tracks for these motor proteins, and these motor proteins basically help carry, some sort of payload along this microtubule track, and they're actually powered by ATP. So motor proteins use the chemical energy in ATP to essentially walk along microtubule tracks within your cells and help carry substances around within cells. Now the centrosome and centrioles are basically like the organization center for your cytoskeleton. We also call this the cell center, and it's made of centrioles, which are a pair of barrel-shaped microtubule organelles. Now newly assembled microtubules radiate out away from these centrioles, and these can also aid in cell division. So while your cells are dividing, the centrioles help to organize those microtubules that aid in the movement of organelles and chromosomes during cell division. And centrioles also form the basis of certain cellular extensions like cilia and flagella, which we'll talk about in the next video. So these centrioles here, remember, are the barrel-shaped structures. Remember, these are the microtubule organization center for your cell. So new microtubules are formed here, and they radiate out away from the centrioles. And you can talk about this as like the cell center, although technically it might not be located in the physical center of your cell. It's the center with respect to your cytoskeleton because your microtubules all eventually link up, for the most part, back towards these centrioles. So in that regard, it's kind of like the center of the whole railroad track system of your microtubules.